everyone, welcome to Dr. Shravani's Med Concepts. Today I am ready with another interesting topic, that is irreversible cell injury, which is point of no return. What happens in irreversible cell injury? When cells are exposed to injurious stimuli, they first undergo reversible changes. So here you can see a healthy cell. When it is exposed to injurious stimuli, it will undergo reversible form of cell injury. But the stimulus is severe enough and it crosses a critical threshold, the injury becomes irreversible, meaning the cell cannot recover even if the harmful stimulus is removed. So this is irreversibly injured cell. Today we will be dealing with this irreversible cell injury in today's session. What are the various causes of irreversible cell injury? Ischemia is the most common cause of both reversible as well as irreversible cell injury. Example is in myocardial infarction. Why is myocardial infarction occurring? Because of ischemia. So in ischemia, what is happening? There are necrosed cells because of ischemia. Here, as you can see in the brown color, this is the injured myocardium. Because of blockage in the artery, which can be because of atherosclerosis, there is injury to the myocardium, which is indicated in the brown color. Another causes of irreversible cell injury are chemical or toxin exposure, severe infections, free radical induced damage, and physical injuries like trauma, burns, or radiation. I have made another video on exclusively causes of cell injury. You can go through that if you want. What are the various mechanisms of irreversible cell injury? One is membrane damage leading to leakage of enzymes. So here you can see the membrane is damaged, because of which the enzymes which are present in the cytosol are released outside now. And there is influx of calcium because of the damaged plasma membrane, which activates various proteases, endonucleases, phospholipases and ATPases. So here you can see the increased cytosolic calcium activates phospholipases and proteases leading to membrane damage. It activates endonucleases causing damage to the DNA and activates ATPases causing ATP depletion. Another mechanism is it leads to the accumulation of reactive oxygen species. Various causes leading to reactive oxygen species accumulation resulting in oxidative stress also leads to irreversible cell injury. So here you can see because of ROS there is irreversible cell injury causing cell death. And organelles are damaged in various forms of injury leading to mitochondrial rupture and leak of lysosomes. So when there is lysosomal membrane rupture, there is leakage of hydrolytic enzymes from the lysosomes causing cell damage. And the last one is there is structural protein damage and DNA breakdown. So here you can see there is structural protein loss and DNA breakdown causing cell death. What are the various hallmarks of irreversible cell injury? They are membrane damage, calcium influx, permanent mitochondrial dysfunction, and nuclear changes. We'll learn about irreversible cell injury changes under these headings. When there is ATP depletion associated with membrane rupture and nuclear destruction, it leads to point of no return or irreversible cell damage. Membrane damage is the first hallmark of irreversible cell injury. Here you can see a cell with damaged membrane. So when there is damage to the plasma membrane, what happens? There is loss of plasma membrane integrity, because of which calcium and water enters into the cell. And there is massive leakage of enzymes from the cytosol out into the serum. So elevated serum markers like creatine kinase, troponins, AST and ALT are elevated, which can be used as markers of irreversible cell injury in certain conditions. Then there is mitochondrial membrane damage. In mitochondrial membrane damage, because of which there is mitochondrial permeability transition pores opening leading to decreased ATP, and there is release of pro-apoptotic proteins which are released outside, initiating apoptosis, and there is lysosomal membrane damage, which leads to release of hydrolytic enzymes out into the cytosol, causing destruction of the cell. Now coming to calcium influx, which is another hallmark of irreversible cell injury. Normally, extracellularly calcium is present, but intracellularly calcium is present at very low levels, because if it is present in excess in the cell, it activates so many enzymes. Hence, intracellular calcium levels are maintained at very low levels. But with the membrane damage, what happens? There is large opening in the membranes which leads to free inflow of calcium into the cell. Hence, there is increased cytosolic calcium, because of which it activates various phospholipases, proteases, endonucleases and ATPases, which worsens the cell damage more. So here you can see extracellularly calcium is present usually, but because of increased membrane permeability or membrane damage, this calcium enters into the cell. Now there is increased intracellular calcium. This increased intracellular calcium, it activates so many enzymes like phospholipases, proteases and endonucleases. It acts on the mitochondria and lysosomes. In the mitochondria, it causes depletion of ATP because ATPases cause destruction of ATP. So there is decreased ATP levels and increased reactive oxygen species, which causes cell injury. Next hallmark of irreversible cell injury is nuclear changes. There are sequential nuclear changes occurring in irreversible cell injury. First is pycnosis, followed by karyorrhexis and then karyolysis. You should remember these nuclear changes in this order only. That is pycnosis, karyorrhexis and karyolysis. In pycnosis, there is condensation of the chromatin into a small dot-like structure. In karyorrhexis, the condensed chromatin is broken down into small fragments like thing. In karyolysis, there is complete dissolution of the nucleus. So, pycnosis, karyorrhexis and karyolysis. So, in pycnosis, there is chromatin condensation. In karyorrhexis, there is nuclear fragmentation. And in karyolysis, there is nuclear dissolution. This pycnosis is characterized by shrinkage of the nucleus, which becomes a small, dense and dark basophilic structure. Microscopically, it is seen as round basophilic dot. In nuclear fragmentation, there is fragmentation of the pycnotic nucleus into multiple small basophilic pieces. In the dissolution, what happens is the nucleus is dissoluted due to the action of DNases and RNases, and the entire cell will be appearing eosinophilic only, and you will not see nucleus because it fades and disappears. So in karyolysis, you will be able to see only eosinophilic mass, but in karyorrhexis, fragmented nucleus will be there. In pycnosis, condensed chromatin will be there. Coming to another hallmark of irreversible cell injury, that is permanent mitochondrial dysfunction. In mitochondria, two things are going to happen. One is mitochondrial damage and another one is deposition of large flocculent amorphous deposits of calcium. So first foremost is mechanism of formation of flocculent deposits in the mitochondria. So when here because of irreversible cell injury, what happens? There is damage to the plasma membrane, mitochondrial membrane and lysosomal membrane. So when there is damage to the mitochondrial membrane, what happens? The calcium enters into the mitochondria, and in the mitochondria it also combines with the free fatty acids and phosphates to form large flocculent deposits in the mitochondrial matrix. If you remember, I told you in the reversible cell injury there is small flocculent deposits of calcium, which is characteristic feature of reversible cell injury. In irreversible cell injury, there are deposits of large flocculent calcium deposits. The second thing going to happen in the mitochondria because of irreversible cell injury is mitochondrial damage. In the mitochondrial damage, there is opening of mitochondrial permeability transition pores, which progressively depletes ATP, and there is also abnormal oxidative phosphorylation, which leads to increased reactive oxygen species. All these leads to necrosis, and because of the membrane damage, what happens? There is leakage of BAC and cytochrome C, which are the pro-apoptotic proteins, and hence intrinsic pathway of apoptosis is activated, leading to apoptosis. Coming to the morphological changes of irreversible cell injury. Under the light microscopy, what happens is there is intense eosinophilia. Cytoplasm becomes intensely eosinophilic because of lack of or destruction of the RNA content in the cytoplasm, and there is loss of nuclei. I told you the nuclear changes, you should remember in this order: pycnosis, karyorrhexis and karyolysis. Pycnosis is the condensation of the chromatin, karyorrhexis is the fragmentation of the nucleus, and karyolysis is the complete dissolution of the nucleus, and there is disruption of the architecture of the cell. What are the electron microscopy features you see in the case of irreversible cell injury. First thing is there is severe membrane disruption. The mitochondria are swollen with dense large flocculent deposits in the mitochondrial matrix, and there is lysosomal rupture leading to release of enzymes, and you will be able to see myelin figures, which are large phospholipid masses formed from the damaged membranes. Myelin figures is the characteristic feature of both reversible and irreversible cell injury. Coming to various forms of cell death. There are various forms of cell death like necrosis, apoptosis, autophagy, and necroptosis, pyroptosis, ferroptosis. All these three are newer forms of cell death. So here you can see the diagrams of necrosis, apoptosis, autophagy, and pyroptosis. I have made a separate video on autophagy. If you want to have a look on that, you can go to my videos on autophagy. What is necrosis? It is uncontrolled pathological form of cell death which causes inflammation, and apoptosis is programmed regulated cell death which is not associated with inflammation. Among all forms of cell death, necrosis and apoptosis are the most common forms of cell death. Coming to the clinical correlation, in myocardial infarction, irreversible cell injury starts 20 to 40 minutes after ischemia, and I told you because of membrane damage, the enzymes present in the cytosol are released out into the serum, which make them as diagnostic markers. So, if there are elevated troponins and CKMB in post-MI patient, it is used as a diagnostic marker for identifying myocardial infarction. In liver injury, ALT and AST elevation are markers of liver injury. Similarly, creatinine is a marker of kidney injury which is raised due to tubular necrosis. These are the questions framed from today's session, and I request you to solve these questions, give your answers in the comment box and give your valuable feedback. If you like this session, please comment, like, share and subscribe to Dr. Shravani's Med Concepts. Thank you.